He studied fundamental epigenomic mechanisms in plant. He then joined my group in 2018 as a postdoc and is recently promoted to scientist. He leads the population genomic study of industrial relevant yeast and machine learning guided genome-wide association study to improve the tolerance of the strain against the lignocellulosic hydrolysate. Ping-Hun, please take it over.
So thanks for the introduction. And I would like to start by thanking the organizers for having me here to share some of our latest research results. So my name is Ping Hong. And then if you are here in the morning session, you probably heard my name several times from Dr. Whistler's talk. So unlike the pin elements in plants, I actually don't have a superpower. But both the pin element and Ping Hong, we both work on a genome. So I think that's something we in common. So today I'm going to talk about my research, which is how we use population genomics and machine learning analysis to uncover genes that are associated with stress tolerance. And we further use those kind of genetic variants to engineer better Escherichia entera strain for industrial use. So although I don't have a superpower, but I think this bug is more like a super bug. So when we see this kind of a barrel of crude oil, I think we usually or often associate it with the fuel. However, there's a certain amount of the oil is actually used for the chemical and materials used in our daily life, such as the nutritional supplements, plastic, detergents, paint, and actually organic acids serve as a building block chemicals, make up a lot of those chemicals or materials I just mentioned. So to actually reduce the use or the dependence on the fossil fuel, we actually need to find alternative way to synthesize those kind of organic acids. And I think that using the lignocellulosic biomass to produce the organic acid serves as a promising alternative way. However, there are many hurdles we need to overcome first. So first, during the pre-processing, there are several lignocellulosic inhibitors that might occur, including the phenolic compounds, HMF and furfural. And then during the fermentation, while the product is accumulated, the pH drops, and there comes the low pH stress and product toxicity. And finally, in the product separation, during acidification step, there's another, again, low pH stress. So from the initial TA analysis, it indicates that using a multi-stress tolerant species can further reduce the costs of a biosynthesis of organic acid by 20 to 30%. So the question is that, is there such a species existed? The answer is yes. Actually, there's an emerging industrial yeast species called Escherichia entera, which has been used in the companies such as Cargill, Jibo, and Lygos for synthesizing organic acids. This species has 5 chromosomes with a genome size about 11 million bases, and usually they're diploids. And this species has been used in the food processing industry or this kind of by human for hundreds of years. And then usually they can be found in this kind of fermentation of cacao beans for chocolate production. And it also has this kind of high glycolytic capability, which is a great industrial trait. More importantly for us, it also shows this kind of multi-stress tolerance, which is a superpower. So first of all, you can see here is a switchgrass hydrolysate without any dilution. And this species actually can grow it happily inside this hydrolysate and use up all the glucose within 12 hours. This species also show very robust low pH tolerance. As you can see here, even under pH 2, it still show very similar growth curve as compared to a control condition, which is pH 5.3. And previous study and our preliminary study also showed this species has very strong tolerance for organic acids. Here, I'm just showing that even under 80 grams per liter of citronellate organic acid, they're still pretty happily growing. So finally, since it's have all this superpower, it also has something we don't like it to have it. So here is the species also show this kind of azole fungicide resistance. And the azole works as a fungicide to kill or to inhibit the fungal growth by inhibits enzyme called ergo-11, which will synthesize the ergosterol for the membrane. And Escherichia entera is known to have the protein ergo-11 have low affinity for their azole. So that's why it is intrinsically tolerant to fluconazole or azole tolerant, azole fungicides. So as an emerging workhorse for the biotechnology, but also a potential opportunistic pathogen, little is known about this species, especially mechanisms under this kind of multi-stress tolerance. So it's very hard, especially when you try to study this kind of things from scratch. So we borrow the idea of like plant or animal geneticists while they use this kind of forward genetics. So we first need to collect enough like natural variants that shows like dramatic or like enough variation of the phenotypes. And then we will perform this kind of genome-wide association study. And our goal is to identify genetic variants that we can use to engineer beta strand with this kind of property. So as an initial like project, we focus on two traits. So first trait is we want to reduce the fluconazole resistance of this species for the biosafety concern or this perspective. But secondly, we want to increase the HMF tolerance of this species for the sustainability purpose. So I will shortly, like briefly talk about the characterization of genetic variation of those strands. So we totally collected the 170 strands, and then 4 of them, we actually did a PacBio, and the rest of them, we just did the Illumina resequencing. So in summary, we identified about 300,000 SNPs, and then actually more than half of them are located on the intergenic region. In previous study in Saccharomyces cerevisiae population genomics, people found that the wild Saccharomyces cerevisiae isolate tend to have lower heterozygosity. So we want to test if we can see this kind of phenomenon in the Eastern Enteros. And here's what it is. And we couldn't see any correlation between the heterozygosity with the environmental habitats. So same thing for the genetic clustering. We try to overlap or correlate the original environmental habitats with the gene clustering, and we couldn't see any clear distinct pattern. So this data or results might suggest that this species may be a nomadic species. They might just like circulate it in different environmental niche. And then if you think of like, oh, it has this kind of super tolerance ability to different stresses, so it makes sense. It probably can survive in like variety of the environment habitats. So we also check the ploidy variants, and then by performing the flow cytometry and gene sequencing, we found all of them are not haploid, and then 75% of them are diploids, and then the rest of them are triploid or more ploidy. And then we also checked into the segmental aneuploidy, and we found 12 strands have this kind of aneuploidy. And then the aneuploidy size can range from like a few kbs into like whole chromosome. We also checked the gene copy number variation, and we found that about 40% of the genes show this kind of amplification. That means they gain one more copy as compared to or at least one more copy as compared to the reference genome. And about like 60% of the genes also show this kind of deletion or reduction, is like partially or complete loss as compared to reference genome. Finally, we also evaluated the loss of zygosity, and we found they are all distributed or more enriched on chromosome, which might be associated with the chromosome recombination. So now we have this kind of abundant genetic material. The next thing we have to do is to check the phenotypes. So the phenotype, my colleague Yusuke Sasaki actually set up this kind of high throughput phenotyping by using ECHO, liquid handler. So we transfer about like 20 to 25 nanoliter of isolates into the plates, and then we measure the size of the colony as like a approximate for the fitness of the cell growth under different kind of stress condition. So Yusuke actually carry out like 3 replicates for each stress condition, and then we can see they show pretty good or robust correlation between each replication. And he also performed like 2 biological replicates in each plate. So now we have all the genotype and phenotype results. The first case I did here is to see if we can find any genes or genetic variants that is associated with HMF tolerance. And indeed, we found there's one, or actually 2 SNPs that are associated with a gene called gene 305. And then one of the SNPs is on the coding region, which causes a non-SNPs mutation. And then the other one is on upstream, maybe the regulatory region. So the pretty straightforward idea is that if we knock out this gene or overexpress gene, what happened in terms of the impact on HMF tolerance? And here is what it is. So we found that by overexpressing this gene 305, we can further enhance the HMF tolerance of this superbug, even into like 4 grams per liter of HMF. And luckily, or something we want, is that by overexpressing this gene, we don't interfere the fluconazole tolerance. And that's what we see in the bottom. So even knockout or overexpression of this gene 305, we didn't see any changes for the fluconazole tolerance. However, when I tried to do the same research for fluconazole analysis, I didn't find any single significant SNPs or indels. And it could be due to our sample size may not be big enough. Secondly, it could be is a polygenic effect. So to tackle this kind of polygenic effect, we set up or build up this kind of pipeline called Mastro. And it stands for Machine Learning Assisted Engineering for Strain Tolerance Rational Optimization. It's pretty long, I know. So actually, this kind of approach has been used in a lot of like human complex disease results. So in short, with the input data, which could be SNP indel or gene copy number variation, and go through the GWAS or association study of a feature selection. Here we use a block HSI lasso, and then go through the machine learning model. We get a model and then do optimization to see how could we change the fluconazole tolerance and the HMF. So here's the result look like. So from the table, we know that, okay, here the first column is the little SNPs where you want to change. And then here is a predict outcome of changing that SNP. And then finally, it's like, is this SNP associated with any genes? And we found there's might be a single mutation that we can simultaneously decrease the fluconazole resistance of this species, but also increase their tolerance for HMF. So by inspecting the genome, we found actually this gene 2301 has another paralog of gene called gene 2300. So it's just like upstream of this gene. So to avoid any kind of gene redundancy, we use CRISPR technology to validate the impact of both genes. So we found that by knocking out either gene 2300 or gene 2301 or double knockout, these strands become more susceptible to fluconazole as we expected, although it's not very significant as we predicted. And most strikingly is that, yep, sorry about the animation, it's a bit slow. Yep, most strikingly is that you can see that by knocking out these genes, we see significantly growth of these strands under 6 grams per liter of HMF, which is pretty striking. So as a conclusion, so I hope I have convinced you that we have provided like important and then very useful fundamental resources, either like genotype phenotype for study strain care intelligence for the community. And then actually this data has been used by another collaborator to study a low pH tolerance. Also, we build up this kind of Maestro pipeline, which can help us systematically to identify and prioritizing genetic variants for engineering. So finally, I want to say that this kind of science needs a collaboration and this kind of community efforts. So JGI here, with the capability of DNA or like sequencing and also DNA synthesis, I think we can be a hub to bridge this kind of research. All right, so I think if you're interested in this kind of research, please contact Yasuo. And then I think currently we are working with Dr. Ian Wilden, working on the species called K. marcinus. All right, so I want to thank everyone for staying here still. And then thanks for the funding agency of DOE and then going through the biosystem design and KEBI and JGI. And I also thanks the people who helped me, like Yasuo's group, people helped me a lot with the experiments. And then Dr. Huimin Zhao and KEBI provide us a lot of like engineering tool. And Dr. Chris Hittinger gave us 7 strands for the genome sequencing. And Dr. Ipe Lee helped implement the machine learning pipeline. Also, I want to thanks the people from the JGI fungal groups, especially Andrew and Hsieh and Vivian. Thanks. I will take questions if I have time.
Great talk. Thank you very much.